Hello, this is a tutorial video of how to use the interactive notebook for Bridgeport. Today we're going to be trying to build an analog structure based off of a known experimental structure, the D2 receptor bound to risperidone. So we're going to be trying to build the analog, basically replacing risperidone with an analog called ziprasidone. So this is a pretty challenging example, so I can kind of show you all the features. And so the first thing that we're going to need to do is make sure that we're set up to actually get all of our data. So I am now in my projects directory, which I have a D2 receptor directory. The first thing I'm going to want to do is make some of my input stuff. So I'm going to make a crystal PDB directory for my crystal. I'm going to make an OPM directory, and I'm also going to make FASTA directory, and the last one is an input JSON, and then I like to make a Bridgeport directory inside of there. So now we have all those things prepped and ready to go. Let's grab our PDB from here. So I'm going to grab that, and I'll throw it in my input PDB right here, right? I'm going to be running all of this on my local. I don't expect any of you to be running this on your local. You'll probably run it on a remote computer. But I'm going to be running it on my local for the sake of this example. So now I'm going to grab my OPM file here. That's not what I want to do. I want to open this in the folder. Okay, there we go. We'll get that out and go back to my D2, go to my OPM, and drop that in there. Alright, so now I have my input structure, my OPM structure. You only need to get one OPM structure for each receptor. You don't have to do it for each ligand, but because you can just reuse it over and over. So that's all we need in terms of input structure. So here's our notebook. We'll run our import statements. The only things you need to change are anything where it has specified input. It has documentation telling you explicitly what to change and what should be there. But that's okay. So I'm not going to go through the documentation. I'm just going to kind of zip through the notebook, and then you can figure it out as need be. So my project directory obviously was called D2, so I'm going to change it to that. This is the same. I've created that crystal PDB. The actual name of my input is 6CM4, and now I need to figure out what chain it is. And so if I open up Chimera, if I open that file in Chimera, here I'll close this quickly for you here, and just open it again. So if I go to Crystal and then open my 6CM4 file, you can see that my D2 receptor is chain A. So I'm going to change this to chain A, and then my ligand name, I'll just always keep the same 4-letter code. My residue name of my ligand is 8NU. You can see that's the small molecule in there, so I'm going to make that 8NU. And then my SMILE string, I'm just going to grab from PubChem. So I'm going to copy that and then replace that. All right, so I'm going to run that. There's nothing in this cell. It's just setting everything up. It doesn't like my Z's, so there we go. Okay, that's just going to regurgitate what you already told it. The first step is now we're going to align our input structure to that OPM structure that I just threw in there. So 6CM4 is the name of that OPM structure. It's always chain A. I don't know where all these Z's came from. And so you can see that now those two proteins are aligned pretty well, actually perfectly, because they're the exact same protein. So that worked great. Now we're going to build our analog. So I'm going to name my analog ziprasidone. I'm going to grab the SMILE string. And replace the SMILE string argument here. From add atoms, this should just be none to start, and then we'll be able to kind of see what's going on. So on the left is going to be our analog structure. So this is actually ziprasidone, and this is risperidone on the right. So you can see that Bridgeport using RDKit is automatically detecting that this ring is equivalent to this ring. Well, if you just look at the visualization, that is not true. I want this ring to be represent this ring over here, so that we can kind of keep this nitrogen, and then this like ethyl linker, and then also, you know, this aromatic ring over here to the left. So we're going to have to basically program all of the maximum common structures ourselves. Okay, sorry, I just had to take a quick break there. But as I was saying, we want to, basically what I had is I had this as none. We don't want those rings, so I'm going to replace it with the atom indices for this ring and get rid of all of that. Now, okay, so now what I want to do is start adding atoms. Like, for example, I want to start at this nitrogen. I want this atom 5 to represent atom 24 over here. So you just add it as a 2D list with the atom indice of your analog and the atom indice of your reference. So it's kept nice and simple where it's left, right. Okay. And then you can see it's going to match my left to my right. So you can kind of continue this process by doing atom 6, which is down here, and then atom 4. So there we go, and now you can see that is working. I already have a bunch of these already defined already, so I'll just kind of skip to that. So, you know, I've worked my way through a lot of this. Obviously I'm missing that nitrogen, so maybe we can just do 20 and what is it, 25. Okay. You know, so I have a ton of this, and let's see if that's enough, right? That might be enough to just kind of represent what we need. Obviously, if you're doing this for real, you can be more thorough and do this entire ring, and but just for the sake of the example, I'm going to see if that's good enough for me. So in this next cell, you can actually visualize how good your alignment is. And so you can see here, everything actually looks pretty good, besides this ring, which is flipped around. So I do need to match this ring to this ring right here. So, okay, I will do that. So a common issue I've seen is if you try and run this cell again, it will work. Okay, I thought it didn't work, but never mind. I guess it does. It might be that this cell doesn't work anymore. Yep. This cell doesn't work. And so if this Jupyter Notebook is meant to be ran sequentially, so from top to bottom, you're not supposed to back around or, you know, kind of bounce back and forth, up and down. That will really screw things up. So if you ever kind of run into a weird error like this, the best thing to do is just select the cell that you want to actually run, and then hit kernel, and then restart and run up to selected cells. That will dump everything in memory and build everything from scratch. And so, see, there you go, works just fine. Not an issue with the code, or just, you know, just kind of a user error. It happens with notebooks. So go ahead and just give it a quick restart. So I need to, you know, kind of finish this alignment here. So I'm just gonna very quickly do that. So we have 22 and 11. We have 23 and 16. We have 24 and 15. We have 25 and 14. 26 and 13. I guess I'm going to end up doing the entire thing. And okay, it didn't like that. I must have messed something up. Let's see. Oops, yeah, I really didn't like that. Alright, let's see. Let's give it a restart, see if that'll fix it. Nope, still doesn't like it. Okay, there we go. Oh, it's because I had 32 instead of 23. I didn't know what I was talking about. There we go, okay. So, yep, that should be pretty sufficient. We're missing 13 and 17. I don't know why. Might as well just add it for the sake of it, since we're going to do all of them. So that's all the maximum substructure that I can add, pretty much. Okay, so now let's check again the alignment, and now my alignment is essentially perfect. So we are all good. All right, so now let's continue. The next step is we're going to separate our ligand protein. Don't need any input. Just run that cell. Next step is we're going to repair the protein. So this one's a little bit tricky. The first input is tails, and so what that means is basically when your PDB fixer is actually going to build, it's going to add your C termini and your N termini to match the wild type, and those will just result in really long disordered strands that we don't necessarily need to model. So I will set my tails to be exactly what's in this crystal. So it's going to be 35 and 42. So 35 and, oh, what was that? 442, I think, right? There we go. That's my last residue there. It's very important that you keep the tails the same for every structure from here on out now. So you might have a different crystal PDB, and it could have, slightly different end, residues that are actually defined in that crystal based off what they decided to solve. But it's very important that you keep the tails the exact same. And so that means that in all your simulations, you have the exact same number of residues all the time. Okay, so just to reiterate, even if I have another crystal PDB or a cryo-EM or whatever it is that has different starting and end points, I want to keep this as my tails argument all the time. Okay, and it'll work out just fine, I promise. So loops is for optimization. I'm going to show you why we're not going to need to do that here in a minute. My gene ID for the dopamine receptor is actually that. And then I need my sequence. So I can go to UniProt. I already found my dopamine human receptor. I'm just going to click on the sequence here and copy that sequence. And then I will replace, that didn't copy. All right. I will replace that sequence. There we go. All right. And those Z's are brutal. Okay, so now it's going to repair my protein. You're going to see something really screwy here in a second. You just give it a minute to run. Well, first of all, you're going to see that my chimera is still in there, which is not good. So, this part is just an enzyme that they stuck on there to stabilize it for crystallization. You can see that it says this right there. So there's a second protein. And it says that second protein is from residues 1002 to 1161. So the easiest way to get rid of that is we have this argument for engineered residues. And so I'm just going to say, okay, get rid of all those residues. And I think it was 1002 to 1161, right? So make that my range. I believe I'm going to have to run this again. Okay, because I don't think this cell is meant to be ran twice if you have engineered residues. Yep, so see now it'll tell you that, okay, it got rid of all these residues, which these are different. These are the MD-Traj indices. They're not the indices in your PDB. And you'll see that, okay, we got rid of that part, and we chose to use a homology model to build it, which creates this like huge intracellular loop 3 that's just not really physiologically realistic. So the best way to, when you have really long sections that you got to fill in, Modeler does not do a good job of that. It's not AlphaFold. So what we're going to do is use a secondary structure, which is an AlphaFold structure. So I'm going to close that, and I'm going to open, not open, sorry, I'm going to use the tools, structure prediction, AlphaFold, and then I'm going to use a UniProt identifier. So I already have that up. Okay, there we go. And let's see, where did I need it? Camera. Here we go. Okay, and then just hit fetch, and that will get you your UniProt structure. So see, AlphaFold's predicting that we actually do have some structure in here, so we want to model that accordingly. And so we are going to save this as a PDB. We're going to call it AlphaFold D2DR, and use untransformed Amber naming scheme. I'm just going to save here real quick, and then we'll move it. So I saved it to here. We'll cut that out. Go back to my project directory, and then I'm just going to throw it in my crystal. You can put it wherever you like, as long as you have the path for it. So now, if I go to crystal, and then I can get that file. Okay, perfect. So my working directory is going to be here. I think, let's see, how do I want to specify a path? So you can specify a path to that with this argument called secondary template. So I'm going to use the path joiner. I'm going to do input dir, which is my dopamine, and then specify my, is it crystal PDB or crystal PDB? It's crystal, okay. Just make sure, and then AlphaFold d to dr.pdb. All right, and so now if I kernel, we're starting to run up to that cell again. Just give it a second here, and it will build something that actually looks a lot more like our AlphaFold structure and won't have that super crazy tail, or not a tail, but an intercept over 3. Okay, so there we go, and that's the protein all fixed, and it has a wild type sequence and is exactly what we want. So looks great. Man, those are really long, TM6 and TM5. Okay, the next step is building the environment. I have the actual executable commented out. This takes a tremendously long time. I'm not going to do it for the sake of this tutorial video. You're more than welcome to do it if you want. I would recommend just not doing it. Make sure that you run this cell so that you add the correct things to your input params, and it'll be in your JSON file later. Okay, then we're going to prepare the ligand. Most of these defaults should be just fine, left how they are. You can always check that they're fine. Make sure that the ligand here is what you want. The ligand here is what you want. I don't see any bonds that are messed up, or we don't have any stereochemistry to worry about. So I think everything is okay. We want to make sure that, yep, this nitrogen is protonated there, and this nitrogen is not protonated there. Okay, looks great. And then the last step is to actually build the force fields. This also takes a really long time, so I'm not going to run that. I would recommend only running that on the terminal and not in this notebook. The point of this notebook is just to build the JSON file, and then you can actually use that JSON file to just run Bridgeport on the terminal. So I'll kind of show you how that works. So first of all, it's going to change. Let me make sure this is correct because I just updated this. Yes, I think that should be right. Okay. And then I'm going to write that JSON file, and let me just, make sure that these are different small strings. Okay, good. That's been a bug, so just want to make sure that was still working. So now you can see I have this JSON file here. So I'm just going to copy that path, and then if I go to my terminal here, get Bridgeport, and then I can just run Python run Bridgeport. Oh, I forgot I moved it to here. Okay, run Bridgeport, and then I will just copy that path. And now, after I activate my conda environment, it's going to run Bridgeport just fine. And so this will take a decent amount of time, but that's and that's okay. So I'm going to run this interactively. Obviously, the one thing that you would need to change for an analog file is I'll cancel out of this. I'm not going to run it for the full sake of the example, but you would need to change the number of conformers. So if we go all the way down to our analog, it'll be number of conformers. If you are running an analog, this needs to be set to 100. And now you have a JavaScript that's entirely ready to go. And just make sure you're running it with a GPU. For an analog, if you're just building something straight from a crystal, you can just run on CPU. It'll take like 20 minutes. If you're running an analog, make sure you're running it with a GPU, and it'll take anywhere from 3 to 6 hours because it'll build 100 conformers and then minimize all of them and choose the lowest energy structure for that. So that's how you do it. Thanks for watching.